Okay, here we go. So we've got finally something to work with. Okay. Let me see if I can do the slideshow for the full screen. Yeah, there we go. And I can even do the highlighter. Okay, so we talked about bright field microscopy. We talked about dark field phase contrast DIC. So what you have to remember about those. Bright field is the most common. Dark field is for smaller specimen, the thinner cells, that allows you to produce highly contrast image of the living cells, although it doesn't reveal you a lot of internal details. Phase contrast gives you practically the 3D image of the living cell. So you don't need to stain its high contrast image of the living cell. And differential interference contrast is, in terms of the purpose, similar to phase contrast microscopy. Now the fluorescence, okay, so you have to use the fluorescence stain to produce an image. Okay. So in this case, fluorescence stain is either specifically attached to certain types of cells, and we will see how it works when we will talk about the identification of microbes, or it can be just non-deliberately staining all kinds of bacterial cells. For example, for instance, there's a stain produced by the company Hearst that stains all mycoplasma. For some reason, it is taken in only by mycoplasma bacteria, and it's used to identify mycoplasma in cells, in clinical samples. It's very convenient. Confocal microscopy, what you really have to understand, it takes practically the slices of the sample and then combining different focal planes and then combining those slices to render the 3D image of the specimen. And finally, two-photon microscopy. At this level, honestly, my knowledge of physics of microscopy is over. I don't really know the mechanics, how it works, but two-photon microscopy helps you to observe thick specimen, okay? Bacteria in the tissue slices, in the skin, things like that. Now, what if we cannot see the microbe using the light microscopy? And I want to ask you which microbes you cannot see using light microscopy. Do you remember? Viruses, that's right. So how do you visualize them? For this, you use electron microscopy. Conceptually, electron microscopy is not very much different from light. And what I mean by that, if you think about this, what is microscopy? You shine the beam of light through the specimen. Does that make sense? Now, if the specimen, the part of the specimen is more dense, how would you see it? Darker or lighter? If it's denser, something is in the way. Darker, yes, it's going to be darker. So darker spots in the specimen correspond to something denser. Does that make sense? Compared to the background, right? Same goes for electron microscopy. You shine, well, it's not shining. You project the beam of electrons either on the surface of the specimen in the scanning microscopy or through the specimen in the transmission microscopy. Do you see the difference? So in transmission microscopy, electrons, which are tiny, tiny particles, as they fly through the specimen, they get refracted by different structures of the specimen. Does that make sense to you? Think about it. Does that make sense? So they get refracted and then they are captured by the detection device, and based on the angle of refraction, right? Based on the angle of refraction, the computer can restore the image. Does that make sense? Okay. Now, in scanning microscopy, essentially use the reflection of electrons from the surface. That's why it is called scanning. So scanning microscopy allows you to see the details of the surface of the specimen. It doesn't let you to look inside though. For instance, here in this picture you can see the cells of the gram-negative bacteria, intestinal bacteria called Campylobacter jejuni, and you can perfectly well see the shape of the cell, right? But you cannot say what is inside. On the other hand, here on top, you can see the image of the Ebola virus, okay? And actually with a higher magnification, you would be able to see the content, what's inside the virus capsid here. But you have no idea how it is shaped. So essentially, transmission gives you a cross-section, cross-sectional longitudinal section, but doesn't let you to render the shape of the object that you look at. Does that make sense? You see the difference? So scanning microscopy gives you a shape of the specimen, but doesn't let you to look inside. Transmission microscopy lets you look inside, but doesn't give you a shape of the specimen. Okay? We good with this? Understand that difference. I may actually show you pictures. Okay? And that's probably the only time I can show you pictures that were not in the PowerPoint. So you have to look at the picture and tell me, is this transmission or this is transmission or scanning electron microscopy? Okay? Any questions? So what if the specimen is, how should I say, too bland and doesn't really stand out from the background, it's not really high contrast. What do we do in this case? In this case, we stain the specimen. One of the things is staining the specimen usually kills it. It's number one. So before staining, we often do another thing. We fix the specimen. But let's take it step by step. So the easiest way to do the microscopy is to do the wet mount. Who has ever done the wet mount before? Okay, so what do you do in the wet mount? How do you do that? Good. You put the drop of water or whatever solution you use, okay, and then you put your specimen in the water, you cover it with a cover slip, and you look at it, right? And if it's a water and you have a sample of, say, a bacterial culture, bacteria will stay alive and you can see them. They're going to be alive. I mean, there's not very much to appreciate about them being alive, to be honest, okay? But water is going to move, bacteria is going to move, and it makes it a little harder sometimes to identify the shape and stuff like that. So that's why we fix the sample. We fix the sample to kill it, so it doesn't move anymore. Does that make sense? There are two ways to fix the sample. You can fix it using heating. You can see it on the picture on the left-hand side of the slide, or with different chemicals. We're going to be using heat. And we're not going to use the heating plate. No way, it's just too much trouble. Much easier is to fix over the Bunsen burner. So flame, you have your cover slip. Cover slip goes through the flame 1, 2, 3, and the sample is fixed. It's pretty simple. The only thing, if you a little hesitant and do it too slow, then you're gonna burn the bacteria. They're gonna explode. Cells are gonna explode, and it's going to be a little messy, okay? But it's all experience. We're going to do it. It is really fast and simple technique, so you can do multiple tries to get a good fixed sample. Does that make sense? Now, once you fix the sample, what you're going to do next? Bacteria, as we figure it out, what is the main component, chemical component of bacteria? Huh? Water. Yeah, it's water. And if you have water in the water, appreciate that image is gonna suck, right? It's not gonna be high contrast and stuff like that. So for this to increase the contrast, okay, we use staining, right? There are two major types of stains. Let me see if I can do the text here. No, I don't think I can do the text. So stains can be, I will try to write it down like this, simple, simple stains, they stain all the microbes the same color. For instance, if you're curious, you can look in the cabinets with the materials and stains for the lab, and you will see a box there with a lot of little bottles. There's going to be a stain called crystal violet. It stains all microbes blue. No matter which microbe it is, it's going to be blue. That's simple stain. Okay. There's a stain called safranin, which stains all the microbes red. It's another simple stain. Or stains can be differential. Sorry, I'll say differential. So I'm going to highlight it here, okay, differential stains. So differential stains allow you to distinguish microbes based on different qualities. Does that make sense? So if you have mixed population of microbes, you have more than one species in the sample, you may be able to see those different populations. You may be able to separate them by the certain quality. Does that make sense? Okay. Now, simple stains, okay, can be positive, like this one on the picture number 2. Positive stains, they stain cells. Okay, that's why they're called positive. They stain cells, not the background. And usually the chromophore, so the molecule that actually gives color, is positively charged. Why positively charged chromophore will stain the cell? Exactly, there's negative charge on the cell, so positively charged chromophore will stick to it. Does that make sense? Okay, well, stains can be negative. Well, my ability to write using mouse is pitiful. Negative stains stain background. You can see it here. Okay, staining of Bacillus megatherium. We have a lot of Bacillus species these days. Okay, so obviously the guys that stain background are usually negatively charged, right? Because negative doesn't really stick to the negatively charged surface of the cell. So far makes sense. So simple stains can be positive, staining the cell, or negative, staining the background. Does that make sense? Differential stains, right here, okay? There are quite a few of them, and they can distinguish cells based on different qualities, and we're going to chat about them a little bit more. Who has heard about gram stain? Gram positive, gram negative. That's the example of differential stain. If you, I hope nobody ever heard about Ziehl-Neelsen or acid-fast stain. That's stain to detect tuberculosis. There's a capsular stain, flagellar stain. They are designed, they are developed to distinguish certain features of the microbes. Okay, so let's talk about them. So stains, the positive stains, can be basic, okay, and acidic, based on the pH, right? Basic stains are positive always, because basic stain is the positively charged chromophore that sticks to the surface of the cell. What we have in our toolbox, we have crystal violet and we have safranin. Okay. They can be acidic. Acidic stains, acidic positive stains, are less frequent, because if you think about this, acidic stains, chromophore should be negatively charged, but it turns out that depending on the chemistry of the cell, they still may stain the cell. Like this one, the cell of some water-dwelling protozoa called Spinaloricus is stained positively, okay, with a Congo red. I don't think we have any acidic stains in our toolbox. We do have negative stains, though. We have India ink and we have nigrosin. So those, as we mentioned, stain background, not the cell. Any questions before we move to differential ones? Now, differential stains, Gram. So Gram stain, it distinguishes bacterial cells based on the cell wall structure. Practically all bacteria can be put in one of two categories, gram-positive or gram-negative. And it turns out that it's not just artificial division. It reflects a fundamental property of bacterial cells. It reflects what their cell wall looks like. Does that make sense? You see what I'm trying to say? So it's not something artificial, it's really it reflects the fundamental properties. For instance, in this picture, you can see that that's the population, two populations of cells. The violet, blue stained, purple stained. I don't know. Its color perception is really hard for me as for a man. Blue purple stained gram-positive Staph aureus. Those little purple cells, and pink stained gram-negative E. coli. Why it is so important? Why we pay so much attention to gram stain? We pay so much attention to it. We're going to do a lab on gram stain. Any idea why? Yes. Exactly. There's going to be different treatment depending on what type of bacteria is found in the patient. Does that make sense? And it turns out that the antibiotic treatment that is used to, that stems from the results of the gram stain. Okay, so if you detect gram positive in a patient, you're going to pick one antibiotic. If you detect gram negative, you're going to pick another. What is the difference between antibiotics? That difference stems from the different cell wall structure. Does that make sense? Different cell wall structure, different permeability. Gram negative and gram positive microbes come from different sources even. You will find them in different places. For instance, gram-positive microbes will be found mostly on the skin, in your respiratory tract. Gram-negatives are going to be found mostly in the digestive tract or reproductive organs as a pathogens. Does that make sense? So it is really important feature. And I heard the interview with the chief of the clinical microbiology lab at Stanford University Medical Center. And she was asked, what is the first thing that you do when you get the sample from the clinic, the patient sample? And believe me, it's Stanford. They have all kinds of sophisticated equipment. She says the first thing, we do the gram stain. Because we do the gram stain, it's quick. We can do it in like 10-15 minutes. We call downstairs, we call physician and tell physician, this is what we found. It's mostly gram-negatives. And the physician can start treatment right away without culture results, without 2-3 hour analysis. There's some freakishly expensive stuff. It's quick, cheap, and pretty good for initial determination of the treatment. Does that make sense? Acid fast. It is used to detect mycobacteria. Not only mycobacteria, but mostly mycobacteria. You can see the red mycobacteria tuberculosis on this micrograph on the right. The thing is, the cell wall of mycobacteria is different from all others. That is why they aren't sensitive, should I say, to gram staining. That's why you should employ different method. Okay, and that's a clinically used method to detect mycobacteria. Endospore stain. You can see on this picture cells of Bacillus subtilis. Look at these two cells that I circle at the bottom. You can see that the cell by itself is stained sort of gray, but on the ends of the cells there are little red dots. Those little red dots are formations that are called endospores. I believe you have heard about spores, have you? Which organisms produce them? Fungi. These are not fungal spores. Those are different structures. Those are escape pods for bacterial DNA. If bacteria experiences harsh environment, usually dry environment, they form endospores. Does that make sense? So they package their DNA into the endospore, so when they die, their DNA still persists. When the environment becomes friendly, they start to grow again. Does that make sense? I'm going to give you an example. Do you like Asian food? Some of you do, some of you don't. So, Asian food usually involves a lot of rice. Right? And go to how is rice cooked in an Asian restaurant? In the rice cookers. And once they cook a gigantic amount of rice, they keep it warm, because if you ever try to reheat the rice over multiple times, the taste is awful. It tastes like I don't know what. Okay, so they keep it warm, like really warm, about 50-60 degrees Celsius. Here's the thing. The microbe called Bacillus cereus, it's a relative of the Bacillus subtilis that you can see on this picture. Okay, so Bacillus subtilis is benign, Bacillus cereus is not so much. Bacillus cereus forms endospores, and those endospores can be found in rice sometimes. When rice is cooked, it's wet, it's warm, and endospores at a certain point, they germinate. That's the word that describes when endospores start to produce living cells. So now you have a rice with Bacillus cereus growing in the rice. And it sounds not so frightening, but Bacillus cereus produces toxin. And this toxin causes food poisoning. and this toxin is heat stable. Does that make sense? So when rice is in the bag, endospores don't do anything because it's dry. You add water, endospores become cells. And it was very well linked. The food poisoning from Bacillus cereus, it was linked to the keeping rice at about 50-60 degrees Celsius in the rice cookers in Asian restaurants. Believe me, by the end of this class, you either stop eating anything, just don't care anymore. That's only one outcome. Okay, I had people who were like told me that they're gonna stop eating pork, beef, fish, rice, poultry, ground beef, anything. Okay. Now, another stain is flagellar stain. It's another type of differential stain. You can see that's the Bacillus cereus, that lovely beast. Okay. You can see cells of Bacillus cereus with hairy-like projections that we call flagella that help the cell move. Why do we need special stain for flagella? Tell me. Why do we care about flagella? Okay, they move. Okay, good. So they move so they can spread faster, right? They're more motile. They can get from one place to another faster. Hypothetically, having flagella, would it make a microbe more virulent, more pathogenic, or not? If it spreads faster? Yeah. Yeah. Usually motile bacteria cause more severe disease because they can get from one locale in the body to another. Does that make sense? What's the problem with flagella? Why can't we see it with a normal light microscopy? It's too small. It's too thin. Okay? So how can we see it? If something is too thin, you have to make it bigger. You have to make it thick. So flagellar stain essentially is when molecules of the dye start to stick to flagellum, make it thicker and thicker, and we can visualize it then. Does that make sense? All right. And finally, capsule stain. It's another species of bacillus. Okay. You can see the thin blue halo surrounding every cell in this image. So these halos are capsules. Capsule is like an armor surrounding the cell. Capsule helps bacterial cells in several ways. It may help cells to attach to the surface, or capsule may protect them from phagocytosis by macrophages. Now, if you would think about two species of bacteria, one having capsule and one not having capsule, which one, hypothetically, would be more virulent? With the capsule, because it's not going to be destroyed by the immune system, right? And it will stick to the surfaces better. Does that make sense? So you see, we can identify a lot of virulence factors. We'll try to write it down with a mouse. Virulence factor. Gonna write. Okay. So that's the term that I introduced to you. So feel comfortable next time we mention that. What do you think is the virulence factor? There you go. Second definition is better. So how pathogenic, how much disease it causes. Does that make sense? Everything. So think about this. There's not really a consensus about how should we exactly determine virulence. but let's say Ebola virus. Is it highly virulent? Yes, it causes very severe disease. Does that make sense? Another example, measles. Anyone had measles? Thank God. Thanks to vaccination, nobody had measles. But measles is extremely infectious and disease is pretty bad, especially in children. So measles is the highly virulent virus. Since nobody received measles, ever had measles, I presume everybody received MMR vaccine, measles, mumps, rubella. Okay? So this is the vaccine that consists of the viruses that still can replicate, but they don't cause disease. So those viruses are avirulent, non-virulent. Does that make sense? Bacteria that are on your skin, are they highly virulent? No. No, they live on your skin, but they actually don't do anything. Does that make sense? So, and virulence factor is anything that can increase the symptoms of disease, that can make the disease more severe. We've already learned flagellum can be a virulence factor, right? Capsule may be a virulence factor. Does that make sense? So we're going to learn about more and more virulence factors as we go through the course. Any questions? What you have to know about the stains. You have to be able to distinguish simple and differential. I give you a definition of the stain. I give you a description of the stain. And you have to recognize, is that simple, is that differential? You have to understand the difference between the positive and the negative stain. Positive stains the cell, negative the background. And you have to know the basics for the gram stain, not the procedure, not the protocol, God forbid, no. What is the purpose? How does it work? So it's cell wall structure, okay? Acid-fast stain, that it's specifically designed to stain mycobacteria. Endospore, flagellar, and capsular. Again, when I say you have to know, it sounds scary, but essentially, if I ask you the question, this stain allows you to distinguish two major categories of cells based on the structure of the cell wall. And I give you answers, flagellar, capsular, gram, and endospore. You pick gram stain. Does that make sense? Because nothing else actually makes sense to this question. Does that make sense? Right? I avoid ambiguity in the questions as much as I can. So you will not try to guess what I meant when I asked that question. Right? So I want to, so we're going to chat briefly of how gram stain works. It's actually quite fascinating. So you have, imagine that you have a bacterial sample with some sort of cocci, gram-positive cocci, so say Staphylococcus aureus, and gram-negative bacilli, say E. coli. So what you do first, you fix them with the flame. So now they're dead. We got it? Next you add a very simple stain. It's crystal violet. Crystal violet, as the simple stain, will stain both types of cells blue. There is no difference so far. Does that make sense? Okay, they're both blue. Next, you add a second reagent, which is called gram iodine. It is also called mordant. So what iodine does, it makes stain to stick to the cell walls, better stick to the cell wall. Does that make sense? Cell's still blue. Nothing really changes yet. During the third step, you add alcohol. And what alcohol does, it removes crystal violet that is sticking to the cell wall. One little thing, in the gram-positive cell wall, crystal violet very well attaches to it, so alcohol cannot remove it. Does that make sense? In gram-negative, alcohol washes crystal violet away. So after that step, after the decoloration, gram-positive cells, they remain blue because crystal violet sticks to the cell wall really, really well. Gram-negative cells become colorless. And then finally, during the fourth step, you add safranin, which stains colorless gram-negative cells. Now it stains them red. Does that make sense? I'm sorry. That makes sense. So far, you don't have to know the entire procedure. Once we go through the lab, that's a different story, but that's going to be later, okay? We have quite a few labs before that. So far, what you have to understand is that it kind of shows you how different the cell wall of gram-positive is from cell wall of gram-negative. Okay? So don't worry about, I'm not going to ask you the process of gram staining during the quiz or first exam. No worries. Okay, it's an introduction. Was I clear? So now we're moving on to the process of culturing microbes. You've already had your first sneak peek into it. Okay, you started it yesterday. And microbes, when you culture microbes, it's often called 5I. Inoculation, incubation, isolation, inspection, and identification. So what is inoculation? That's when you put the microbe into the medium. So there are two words that we haven't discussed before. The word inoculation and the word medium. What is medium? Exactly. Anything. Can you think of any medium based on your household experience? Huh? Bread. Perfect. It's a medium for fungal growth. Something else. Any type of food that's getting spoiled by microbes. Are you a medium? Sure. Every time you're sick, you are a medium. You're a medium right now. There's a bunch of microbes growing in you. Okay. So when we inoculate, what is the most? We have to collect the sample first, right? What is the most important part in a sample collection? How should we collect the sample? Sterile procedure. Very good. So our sample collection should be sterile. Yesterday we used sterile cotton swabs. Okay. So okay, sterile procedure. Which clinical samples can you collect? Which clinical samples can you think of? Urine, okay, urine is the clinical sample. Something else. Blood. Huh? It's louder. Okay, throat swab. Respiratory secretion, snot, saliva. You can collect saliva. Come on. Feces. There's one more which is really important, and usually goes unnoticed. It's a fluid. Say? No? What did you want to say? Come on. Semen. You can do semen, yes. Semen or vaginal secretions. You can do reproductive tract secretions. It's a fluid, just fluid. Really important. Spinal fluid. It's very critical because basically if your spinal fluid comes positive, probably you boned. You're in big trouble. Okay? Because that's sterile. It should be 100% sterile. Does that make sense? So those are clinical samples. What about environmental samples? Soil. You can do soil. Dust, toilet swabs, sink swabs, everything, yes. And actually, tomorrow, that's what you're going to do. You're going to be equipped with cotton swabs, and you're going to go around the campus, the room, whatever, and you can sample whatever you want. Huh? Home? You can do it, yeah. Well, the only downside is that you better do it right before the class. Like when you leave in the morning. You can take a cotton swab and then bring it here and put it on the plate. But people sample gym, female locker room. Nobody sampled male locker room for some reason. Just bottom of the shoe, inside of the shoe piercing. One guy, very daring person, sampled his urine. Nobody sampled poop. We sampled urinals. We sampled toilet bowls. We sampled breakers. Microwave in the breakers, elevator, button. I mean, everything that you can think of. There's a lot of stuff, and sometimes the results are quite unexpected. We found some very unpleasantly looking stuff in a shower in a female locker room, in the floor. Pretty bad. So once you collect the sample in a sterile way, that is important. You inoculate the medium. Medium can be different. You already worked with agar. You will see other different types of media, all of them based on agar. In some cases, you have to inoculate live organisms. For instance, helminths cannot grow in culture. You have to put them like in a mouse. Okay. And media can serve different purposes. For instance, the one that you used yesterday, TSA, tryptic soy agar, it's just for growth. Some media can help you to identify the microorganism. Another can help you to differentiate them, selectively grow some. Does that make sense? We'll learn about that. And once you're done with that, you stick it into the incubator. Okay. And incubator is temperature controlled. Temperature can be different, okay? The ones that we have in the room, the only reason I do not take you into the room and show you incubators is because it's not exciting. Cramping 26, 27 people in that space has nothing to do with excitement. And it's pretty much a box that's 37 degrees inside. Some microbes grow at the higher temperature, some grow at the lower temperature. Some need extra CO2, some need no oxygen and whatnot. So it's gas, pretty much atmosphere controlled and temperature controlled. Does that make sense? It may take overnight. I'm pretty sure we're going to get some results today. Okay. It may take weeks. Mycobacterial tuberculosis grows really, really slow. And then, once you get your plate, you look at the plate, you look at the colonies, and you isolate the colonies. Okay. Now you sampled your skin. Do you expect one species of microbes to be on the skin or more than one? More than one, yes. And if you want to figure out what is a specific colony, you have to subculture it. Does that make sense? Imagine if you're planting. I know that some of you are planting. Okay, you've got a bunch of seeds and you don't know what there is. So put them in the soil, and you have two different types of plants. Now you can separate them because you see that those are two different types. Does that make sense? You can physically separate them, put them on the separate patches of land, and then grow them independently. Does that make sense? And once you've got your colonies, you can do inspection and identification. Inspection is pretty much describing how the colony looks like. Is that, what can you tell about the colony? Let's start with this. Which qualities of the colony you can see? What do you think? Have you seen bacterial colonies? No, it's like a little thingy. Okay. Bread mold. What can you say about bread mold? It's, say again? So you can say color.
Usually looks fuzzy.
Fuzzy, appearance. Right? So you can just from looking at things, you can say, tell about appearance, shape, color. And that's enough actually to identify certain things. Okay? And then once you've got the colony, you can do identification. There are different techniques that we're going to talk about. We're going to touch on three basic approaches: immunological approach, genetic or molecular biology approach, and biochemical approaches. Okay. So let's talk about different types of media that you can inoculate. Liquid is liquid. It's usually water-based. Okay. You can prepare the simple liquid media at home. Any idea how? No? No? Liquid media with a lot of nutrients. Beef stock, chicken stock. That's perfect liquid media. Especially if you filter it well and it becomes transparent. That'll support bacterial growth, be my guess. That's actually pretty much what is sold is an extract of beef that you dissolve in water, and that's your media. It will support the growth of microbes. Okay? Why we use liquid medium, we use it either to accumulate large amounts of bacterial cells, so any microbial cells, or we use it as an indicator, okay, to detect differences. Okay, for instance, in this picture in the bottom, you can see 3 samples of bacteria that can ferment sugar. Fermentation. What does it produce? What is the result of fermentation? That's alcoholic. What is the second type? You probably can detect alcohol in the medium. Gas can be a result of both, but there are 2 basic types of the alcoholic fermentation and, like if in human cells there's no oxygen, what's the anaerobic, it's the conditions that it goes, but what's the final result? When your muscles are deprived of oxygen, what accumulates in there? Lactic acid. So you have alcoholic and acidic fermentation. And if fermentation results in accumulation of acid, the indicator, chemical indicator, will change color. And you can say, okay, this bacteria in this medium can ferment sugars. Does that make sense? Okay, so you can detect fermenters and differentiate them from non-fermenters. Right? Solid medium. On the right, TSA, trypticase soy agar. That's by far the most common bacterial medium. Okay. Agar has a wonderful feature. It can become liquid at the high temperature. Right? So you can heat it up, you can autoclave it, pull it out of the autoclave, it's going to be liquid, it's going to be fluid. You can pour it in the petri dishes, leave them on the table, and as they cool down, agar solidifies. Right? Pretty much, when you make jello, that's gelatin. You liquefy it when you heat it up, and then as it cools down, it becomes solid. Can you use gelatin as the medium? Absolutely. The only problem, bacteria can eat it. So you may end up with the bacterial culture slowly eating through the gelatin as the solid foundation for the medium. That's why agar is more common. If you want to do it at home, agar, I told you can buy it at the bakery department in practically every grocery store. For some reason, like a can this big costs $15. I don't know why, don't ask me. You can buy actually ready-made petri dishes from Amazon. And I think it's about $1 a dish. They're sterile, they're good to go. So if you have a kid that's about to do the science fair, it may be a good idea. The only problem is the incubator, but let me tell you, most bacteria will grow at the room temperature, just slower. Much slower, but they will. Okay? So, and I think it even comes with a prime shipping. Now, you have liquid, you have solid. Solid media is used to separate the colonies, okay? So you can see different bacterial colonies. You can see on this medium streaks of the colonies here and here. By the way, this name of this particular medium on the right, it's a charcoal medium. It does contain charcoal. So bacteria require it. I don't remember which one. No, I don't remember which one. I don't want to lie to you. Now there is one medium in between that is called semi-solid. It means that it doesn't have a good solid surface right here. Okay, it is also called a slant medium because when you pour it in the tube, it forms an inclined surface that is called slant. So why do we use semi-solid medium? Remember we talked about bacteria being able to move? Now, in the liquid medium, you cannot really tell if they can move or not because they will just float, right? In semi-solid, you can compare bacteria that are motile and can spread through the medium to bacteria that are non-motile and remain in one place. Does that make sense to you? We will actually do that lab first day next week, on Tuesday. That's the lab that we're gonna do, the isolation technique. Okay. Now, this is the example of special medium called triple sugar iron medium. This is the first example of the media that helps you to identify, distinguish different types of microbes. So look on the left here. Slant is what color? Yellow, right? And there's little, it's lifted from the bottom. How can it be lifted from the bottom?
Bacteria or whatever sample like move down there.
Gas. It produced gas. Gas accumulated at the bottom, lifted the medium. Does that make sense? Why Normally the slant is pretty much this color. Okay, red. So why it became yellow? Because there's a fermenter. It's a fermenter and gas producer. Does that make sense? Okay. This is something that ferments only glucose. There are 3 different sugars: glucose, fructose, and I think mannose. If the microbe can ferment fructose and glucose, media remains yellow. If microbe can ferment only glucose, medium remains yellow at the bottom. Here the microbe produces hydrogen sulfide. And here microbe doesn't do anything, it just grows on top of the medium. And actually on the left is E. coli. It's a gas producer, and it's a lactose and glucose fermenter. Second to the left here is salmonella. Okay. Here, third one is Shigella, and this one Pseudomonas aeruginosa. Using this medium and knowing the source of the sample, you can pretty much identify the microbe right away. Does that make sense? Because if you sample something from the respiratory tract, you expect certain types of microbes. If you sample something from the digestive tract, you expect different types of microbes. Does that make sense? Right? Like if you go to Hawaii, you don't really expect to find a lot of cross-country skiers there. It's mostly going to be surfers. Right? Based on a composition, medium can be complex and defined or synthetic. Synthetic medium is the one with the known composition. So in terms of synthetic medium, what you do, imagine that you take 21 amino acid, known amounts of sugars, vitamins, sodium chloride, calcium chloride, put it in the known amount of water. So you know exactly which chemical components are in the media. Does that make sense? Complex medium is chicken stock. Do you know the exact chemical composition of the chicken stock? No. Trypticase soy agar. It has the soy protein that was partially digested. We don't know how it was digested. So trypticase soy agar, is that complex medium or defined? We don't know how it was digested. It's a complex, yes. We don't know the chemical composition. Now, complex medium, okay, is used to grow microbes with unknown nutritional requirements. Does that make sense? Imagine that you throw a party and you have like 30 people coming, and you don't know what those people like to eat. So you will try to prepare a variety of food so everybody has something to munch on. Does that make sense? Because you don't know what, but if you have somebody, the friend coming, the friend just survives, I don't know, on cupcakes, and that's it. Doesn't eat anything except cupcakes. You're not going to cook steak. You're just going to get cupcakes. Does that make sense? So that's why we need defined medium, to grow microbes with the known nutritional requirements. And we can compare them. If we know chemical composition, we can take one component away. Does that make sense? And see, can microbe grow without that amino acid? Can microbe grow without this amino acid? You see the difference between complex and defined? Any questions so far? We're going to finish with this, and we're going to take a break. Now, purpose of the medium. medium can be of the general purpose. To grow stuff. The TSA that we used yesterday, and we're going to use it today again, it's a general purpose medium. It will support the growth of a wide variety of microbes. Does that make sense? Like everything will grow. Okay? It's typically a complex medium. You already encountered TSA. There's a brain-heart infusion, for example, which pretty much it's a brain. I think it's a cow. So cow brain, cow heart. They ground. There's something done with them. I think they kind of frozen or lyophilized, so they turned into the powder, and you can then dissolve this powder in water and kind of, freeze-dried stock. Okay. Enriched medium. Enriched medium is used to grow so-called fastidious microbes. Fastidious microbes, I sometimes call them picky microbes, because they need some specific component. For instance, there's a so-called chocolate agar. It doesn't contain chocolate. Okay. It contains cooked blood. Sheep blood is heated up, red blood cells fall apart, and release whatever's inside. What's inside the red blood cells? Huh? Hemoglobin. And what's inside the hemoglobin? Iron. The cold iron is master of them all. The microbe called Neisseria meningitidis or Neisseria gonorrhea, they require iron for growth. And the easiest way to add iron to the agar is to just throw in some blood. Does that make sense? So that is enriched medium. If you put Neisseria on the regular TSA plate, it's not going to grow. There's not enough iron there. Okay. Selective. Selective medium contains certain inhibitory agents. So selective medium doesn't let some microbes to grow, so it allows you to identify certain microbes. For example, there's a medium called mannitol salt agar, and I want to focus on the salt. So the agar, the plate that contains a lot of salt. Do you eat salty food? Does anyone do canning of vegetables like pickled, pickled tomatoes or something? Well, why do we salt stuff? Why do we do pickled? Yep, exactly. It preserves. Why? Why do, Exactly. Bacteria cannot grow in a high salt environment, really. Because it's a lot of salt. For most of bacteria, it's going to be a hypertonic environment that will just shrivel. Does that make sense? Well, there are some microbes that actually like salt, if I may say so. One genus that loves it is Staphylococcus. Staphylococcus easily grows at 7.5% salt, which is a lot. It's really a lot. Nothing else grows there. Staphylococcus does. So it's a selective medium. It selects for Staphylococci. Does that make sense? Another example is the EMB agar. It contains certain dyes that support growth of only gram-negative microbes. So gram-positives like Staphylococcus or Streptococcus will not grow. Gram-negatives, microbes that are commonly found in your digestive tract, will. And the last type of medium is differential. So differential medium supports the growth of many types of microbes, okay? But different microbes look different on it. Does that make sense? For instance, the medium called blood agar, it actually contains blood. It allows you to distinguish between the microbes that are hemolytic and non-hemolytic. What do you think hemolytic means? What's hemolysis? Yeah, lysing of the red blood cells. Okay, so hemolytic microbes will form like a halo of completely lysed red blood cells. Non-hemolytic will not have this halo. Does that make sense to you? That's example of, they both gonna grow, but they're gonna look different. Okay? So that's differential. Now, three types of cultures that you can find on the plate. One is the pure culture, when you have only single species of microbes. Another type is mixed culture, when you have more than one species. And finally, contaminated culture is when you want one species, but there is more than one because somehow you got contamination. You don't really want contamination. Referring to our yesterday experiment, what type of culture do you think you're going to get on your TSA plates? It's not, you don't expect one sample, so it's going to be mixed, right? You really don't know what it's going to be. Contaminated, strictly speaking, is, let's say, I have a tube, okay, and I know that this tube has only one species. And I get this, I put it on the plate, and it turns out it's not one. So I wanted one, but it's two, so it's contamination. Contamination is unwanted. Mixed, I don't know. Okay? Does that make sense? Now, before we take a break, so what you have to know, you do have to know three different physical states of medium. Okay? Solid, semi-solid, and liquid. And basic applications. So solid is used for isolation of microbes, semi-solid for motility testing, and liquid can be either indicator media or can be used for accumulation of the large amounts of microbes. Okay? You have to be able to differentiate synthetic and complex. Right? Synthetic, known chemical composition, complex, unknown chemical composition. Question? It's used for motility testing mostly. Okay, and it doesn't have the solid surface. Remember when we talked about slant? Question? Jason? Question? Okay. For complex defined known chemical composition, complex unknown. And for purpose, understand the difference between, say, general and enriched. Know what the fastidious bacteria is. One thing, I saw it, so I'm going to tell you. Fastidious is bacteria, not the medium, bless you. Does that make sense? Fastidious bacteria grows on enriched medium. Does that make sense? Understand the difference between selective and differential. Right? Selective, one type of microbe grows, others are inhibited. Differential, all kinds of microbes grow, but they look different on the medium. Okay? And three types of culture. How these questions are going to look like? Well, first of all, I'm going to give you a quiz. You will see the examples. Second, Something like, I give you definition and you pick the right answer. Does that make sense? Let's take a break.